All right, so part two now. Secondary structure. So your primary structure is just the sequence and is stabilized by your peptide bonds. Secondary structure, backbone hydrogen bond interactions. Okay, now there are three major secondary structures. There's an alpha helix, a beta sheet, and a beta turn. Now the driving force for the formation of secondary structures is your backbone hydrogen bonds. These are referred to commonly as local structures. Now, wool was the substance that William Asbury used to kind of theorize about protein structures and gave some insight specifically into the secondary structure. He studied fibrous proteins using X-rays to determine diffraction patterns. Okay. He showed that there were drastic changes in the diffraction of moist wool fibers as they are stretched. So unstretched showed periodic repeat distances of 1.5 and 5.1 angstroms. So he measured these like, whenever it would be relaxed compared to whenever it was stretched out. The stretched wool showed periodic repeats of 3.5 and 7 angstroms. He proposed that the unstretched protein molecules formed a helix, which he called an alpha helix, and that stretching caused the helix to uncoil, forming an extended state, which he referred to as the beta form. Now, he was the first to propose that main chain hydrogen bonds had a role in stabilizing protein structures. So basically, he said, I think that it's these, maybe he did not refer to them as backbone interactions, but ultimately came to the conclusion that backbone interactions drive or stabilize protein structures. Now, what do you do when you have a cold? Linus Pauling discovered that the alpha helix, even though the structure of the amino acids was known, the structure of proteins eluded description until Pauling constrained the structure, bond lengths and angles of the peptide bond, assuming a rigid planar model for the peptide bond. Pauling modeled two structures, a helical structure or an alpha helix and an extended structure or the beta sheet. He did not require the number of residues per turn to be whole numbers and initially ignored Asbury's numbers. So the characteristics of an alpha helix, here is on the right-hand side here, that's a snapshot of an alpha helix. Now, some of the numbers to describe the alpha helix. The rise per residue is 1.5 angstroms. The pitch or rise per turn was 5.4 angstroms. So basically, if you look at this as a starting point, this goes and it loops and it stops right here. So whenever I say stops, I mean it completes one complete turn. And it takes, that complete turn has a height of 5.4 angstroms. Each amino acid built into this rotation has a height of 1.5 angstroms. So what is accepted today and theorized is that about 3.6 amino acids are required to make one complete rotation. And on average, there are about 10 residues per helix. So if you think about this as a helix has about 10 amino acids, and there are 3.6 AA per turn, that means there's just a little bit over, I'm going to say 2+ turns per helix. So one thing that's worth remembering, and to provide credit where it's due, William Asbury observed that unstretched wool with periodic repeat distances of 1.5 and 5.1 angstroms. So he came with these numbers that are pretty close to what Pauling later concluded. Now, the hydrogen bonding pattern in an alpha helix is important. An alpha helix is a right-handed spiral. The alpha helix is stabilized by extensive hydrogen bonds. The CO, so I'm going to label this right here and this right here as the pair that participate in the hydrogen bond. So these pair the C termini of this amino acid right here and the N termini of this amino acid right here make up a bonding pair. Now, this is going to be the H bond acceptor, and this is the H bond donor. What's worth noting is that, well, we have repetitions. Like this guy is going to be an acceptor, and this is going to be a donor. Then this is going to be an acceptor, and then this is going to be a donor. So you have this complex network of hydrogen bonds that are produced. Keep in mind, hydrogen bonds are non-covalent bonds. They're not particularly strong, but in numbers, when you have lots of them, they get considerably stronger. So the last 4 residues at either end of the helix participate in only one hydrogen bond. We have a model that's pretty well accepted, and that is amino acid, the very first amino acid within that helix is going to be the I amino acid, and then the one that's going to hydrogen bond with is the I+4. So here what we have is, this is our nitrogen, this is our alpha carbon, and this is the C termini of amino acid number 1. And then this is going to be right here, the N termini of amino acid number 5. Now, a helical wheel is a nice way to visualize basically the positioning of different amino acids and how they come together to make a helix. So amino acid 1, 2, 3, and this listing is in the peptide bond. How if you look at these amino acids, where they're positioned, we've got a new color here that shows, this new color represents the new turn. So blue is one, is one complete rotation. Red is the second rotation. Hydrogen bonding is going to take place between amino acids 1 and 5, 2 and 6, 3 to 7, 4 to 8, 5 to 9. And so, as you can see, it just gets more and more complicated. Sorry about that. But this is just a model for showing the pairings of amino acids, or sorry, not amino acids. Yes, the pairings of backbone hydrogen bonds, and which amino acid is going to have this hydrogen bond, and so on. So a dipole moment, an alpha helix possesses a dipole moment because each peptide has a dipole. So there's an overall partial negative charge and a partial positive charge at each of these termini. The amino terminus is going to have a partial positive charge, whereas the carboxy terminus will have a partial negative charge. Now, the significance of this is really just to illustrate what the termini of these are capable of doing. These partial positive and partial negative charges can interact with other things in the environment. So they're going to find things that help stabilize them. Specific amino acids might be nearby. Now, a biochemist can use the alpha helical information to do some calculations. So if an alpha helix has 15 amino acids, how many angstroms would that cover? Well, this is dimensional analysis problem. 15 residues, it's 3.6 residues per 1 turn. This alpha helix has 4.2 turns. Well then, 4.2 turns, how large is this? Well, if it has 4.2 turns and 5.4 angstroms per turn, 4.2 times 5.4 is 22.7 angstroms. So that's how tall that 15 amino acid alpha helix is going to be. Then if you do that 15 times 1.5, which is the rise per AA, it's going to put the height at 22.5 angstroms. 40 angstrom wide membrane. how many amino acids, just to kind of put this into effect, if you have a protein that is spanning the entire lipid bilayer, well, if that lipid bilayer is 40 angstroms, how many amino acids are going to be required? Well, 40 angstroms, 5.4 angstroms per turn, it's going to have 7.4 turns. 3.6 residues per turn means this polypeptide needs to be about 27 residues long. Yeah, again, 27 residues long. All right, so that is where I'm going to stop with this video.